Okay guys, this morning we're going to talk about metabolism. Now metabolism, remember we usually associate metabolism with making energy for our bodies, all right? But there's actually two types of metabolism. I have catabolism and I have anabolism. Now my catabolism, what this means in the shortest amount of words is that I'm going to break down something. And you guys know that if I break chemical bonds, then I get to release energy. So a good example of catabolism would be the respiration process that we're going to start going over. And then anabolism, that's going to be building up. And a process that builds up something that we use for energy is photosynthesis. And we'll kind of talk about why in a later video, why that's a good thing for energy. Now, whenever we talk about energy, there is one molecule that we always talk about. Our energy molecule is ATP. Okay, ATP is always our energy molecule. It's the molecule that fuels all of the cellular processes. So we need to go through what ATP means. All right, ATP stands for adenosine triphosphate. Okay, that's ATP, adenosine triphosphate. Now, what this looks like, I mean, you guys can imagine triphosphate, that means there's three phosphates and then we have an adenosine. And so what we're going to do is we're going to draw this little guy, all right? So let's start out with the triphosphate first. Now remember, phosphate is PO4, so here's one phosphate. And then here's my next phosphate. And then here's my last one. So there's three phosphates. Now remember, when we talked about phosphate a little bit ago, earlier in the semester, we talked about how unstable it was because of all the oxygens. But now look, now I have three of these suckers next to each other. So this phosphate right here, he is super unstable. He is ridiculously unstable because just imagine if you had three magnets that are all the same polarity, like three negative magnets next to each other. They wouldn't want to stay there. Say they'd want to get away from each other really badly. And that's why this PO4 molecule right here, this triphosphate molecule is really, really unstable because each of these phosphates, this guy, this guy, and this guy, they all have negative charges and they don't like to be around each other. That's why here after a little bit, when we talk about how we get the energy out of the sucker, they're so easy to break. Then I'm going to attach this oxygen to this guy. And hopefully he looks mildly familiar. Well, let's look at chemical structure. I have a bunch of carbons, because I know there's carbons at these points. And then I have a lot of oxygens attached to him. That means he is a sugar. This sugar is ribose, right? And the reason I have ribose here is because this guy, as far as sugar goes, we're just going to call him, he's very sticky. And so since he's sticky, I'm going to be able to stick these phosphates to my last molecule, which is going to be my adenine group. And so he's attached to a nitrogen, okay? And then I have a nitrogen here. There you go. So there's my first part. And then I have another NH2 up here. I have an N. I have an N. There you go. So this guy right here, this is where the adenosine comes from. He is adenine. Now adenine, if I look at him, if I look at the elements in there, he is a nitrogen base. And more specifically, since he has two rings and it's adenine, this nitrogen base is a purine. So there's my entire ATP molecule. So I have my adenine, my adenosine right here. I have a ribose that attaches everything, and then I have my three phosphates. And so what's going to happen, like I said, since these phosphates don't want to be next to each other, they break off really easily. Well, out of these 3 phosphates, this last phosphate, he's the easiest one to break off because he's the last one in the row of 3. And so whenever I break this bond right here, so what I'm going to do is I'm going to break the bond between the 2nd and 3rd phosphate. When I do that, that's going to release energy. And that's why this is probably one of the best energy molecules we have, is because the polarity of the PO4s and how they don't want to be next to each other. This is, whenever we make this molecule, this is very, very short lasting. Say it doesn't stay there very long because they don't want to be next to each other. So my next question, how do I get the energy out of this sucker? Well, I want to break it. So let's think about our reactions from the very first unit. What chemical reaction do I do to break something? That's called hydrolysis. Because remember, hydro means water, lysis means to break. So I'm going to break something using water. And what am I breaking using water? It's going to be ATP, so hydrolysis of ATP. And what that equation looks like, so instead of drawing out that big molecule every time, I can just do that, ATP plus water, and that's going to give me ADP. We'll talk about what that is. P with a little I, we'll talk about him. And then, of course, the stuff that we want most of all, that's going to be my energy. Now, going back, this ADP, this is adenosine diphosphate, which makes sense because if I had, here is a simple graphic, if I have 3 P's, and then I add water, and my whole goal is to break that guy off, now I have A with 2 P's, ADP. And then I have an extra phosphate. And then, of course, the energy that I'm talking about. So that's what ADP is adenosine diphosphate. Now this P right here with an I, that I stands for inorganic. And remember, if something is organic, it has carbon in it. Well, if you look at the big molecule up here with my adenosine triphosphate, that has carbon because it has carbon in the ribose, there's carbon in the adenine. But now that I've separated that phosphate, he's no longer a part of that molecule, so now he's inorganic, or he doesn't have any carbon. And then, like I said, the molecule that I want from this reaction is going to be the energy. All right, so that's how we break ATP. Now we need to talk about where this happens. All right, so where do I make the most energy in my cell? Say, I don't like to say it, but where's the powerhouse? Oh yes, our friend, the mitochondria. So let's talk about mitochondria structure. First of all, we know he looks kind of like this, right? So this is my overall mitochondria. This yellow line right here, this is my outer membrane. And then, as we've always drawn, here's that stuff in the middle. So outer membrane's very outside. Then this guy, all this empty space, this is called the inner membrane space. This line right here, we've called it before, this is my cristae. And then this stuff here in the middle, this is called my matrix. So whenever I start making my ATP, it's going to be right here in this organelle, and then more specifically, I'm going to look at these 3 parts, the inner membrane space, the cristae, and my matrix. Because the thing that's going to make my ATP is an enzyme called ATP synthase. And how do I know it's an enzyme? Because it ends in ase. So this is an enzyme that makes ATP. Where he is located in the cristae. And this is a big, because remember, enzymes are a class of protein, and so this protein, this enzyme, is sticking in my cristae. And my cristae, what he is he is going to be a phospholipid bilayer, which makes sense when we talked about endosymbiotic theories and where these organelles come from. He has two membranes. Well, there's the other membrane, is the cristae. So if I wanted to draw ATP synthase, he looks like this. So we're going to draw a box with lines, and then we're going to draw an empty box. So this is ATP synthase right there. And where he is located is, since he's in a membrane, so let's draw some phospholipids around him. There you go. All right, so this part on this side, this is my inner membrane space, and this side over here is my matrix. And so really, if I wanted to draw him over here, like let's pretend he's right here, here's the box with the lines, and then here's my empty box. So you see how this part faces the inner membrane, and then this part faces the matrix. All right, so now what we need to talk about is how. So how does this enzyme, this ATP synthase, make ATP? All right, well, remember, he's an enzyme. So let's draw him again. Here's the box that's lined, and then here's this box. But now I'm going to do two little cutouts right there. All right, let's remember, this is an enzyme. So these guys right here, this is going to be my active site. This is where my substrates come in. All right, well, if I want to make ATP, I just have to do the reaction backwards, correct? I'm going to take ADP plus an inorganic phosphate, and then that is going to create ATP. So if I have this ATP synthase over here, I need my two substrates. So here's one triangle. This can be my ADP. And then here's my other triangle. This can be my inorganic phosphate. And so what they're going to do is they're going to go fit inside these active sites. And then, because enzymes facilitate reactions, that's going to spit out, we'll just call them a big triangle, and this will be my ATP. All right, so that's what the enzyme is doing. He's taking in my two substrates, facilitating the reaction, spitting out ATP. But now we need to know how that's actually doing this. All right, so let's draw it again. So here's my ATP synthase. And then here is the bottom part of it with my active sites, right there, right there. So what's going to happen is this top part up here, this top part is called F1, this bottom part's called F0 or F0. What this guy does is, this guy's like a water wheel, and he's going to start to spin on top. When he starts to spin on top, that's going, so that way, here's my substrates, here's my ADP and my P. When they go fit inside those active sites, we talked about how enzymes will kind of hug their substrates, so that way the reaction takes place. Remember, that's called induced fit. And so when the substrates enter those active sites, this top part, he's going to spin. When he spins, that's going to push these two together, and that's how I get the ATP. All right, so substrates go into the active site, this guy spins. When he spins, that forces them together. I get ATP in the end. All right, so then the next question is, why is that sucker spinning on top? All right, so let's draw our picture again. So there's our F1, and then here's my F0. All right, how I get this guy to spin is actually by protons. And protons, let's write it out here somewhere. A proton is just a fancy word for a hydrogen ion, right? So I have these protons here. Out here in the, remember this is the inner membrane space. And this down here is the matrix. So I have these protons, and maybe I only have 1 or 2 over here. Well, as everything goes, we like to flow from high to low. And so whenever I have these hydrogens and they're like, hey, this is a really, really high concentration over here, and this is a really low concentration. So I want to go from high to low, but this is the channel that they go through. So these hydrogen ions, they start to go through this ATP synthase. When they go through the ATP synthase, that starts this to turn. Remember, it's like a water wheel. So this guy starts to spin. When he starts to spin, then these sitting here in the active site, they get pushed together. And when they get pushed together, that makes my ATP. All right, so we're getting somewhere. I have my enzyme up here, ATP synthase. I know that he's going to work on an induced fit when the 2 substrates come in and I spit out ATP. How I'm going to get the induced fit is by making this top part spin. And how do I make him spin? By dumping a bunch of hydrogen ions through the top of it. So now, I have to ask myself, how did the protons get to the top part? That's because of a newfound friend. This newfound friend is called NADH. Now, NADH, this guy is a great electron donor. He's actually the best one we have. And so the reaction that we see from NADH is that I have my NADH, and then when he comes up to a complex, a protein, it's going to break it into NAD, my protons, which are really important, and then I'm also going to get some electrons, which are going to end up being important here in a second. All right, so that's why NADH is super important to us, is because I can get my protons and now I can get my electrons. And so how that works is, let's draw another picture. So here's going to be a protein complex, right? And then, of course, I'm going to have some, here, let's just do some nice pink. So here's some phospholipids. And then here's my ATP synthase. There's ATP synthase. All right, so what NADH does, here's my NADH. He's going to go up to this guy. This is a big protein, okay? This is a protein pump right here, this white guy. Protein pump. Protein pump. And so when the NADH goes up to him, that protein pump is going to strip him apart into NAD, my protons, and then the electrons are actually going to stay in here. All right, so now if you noticed, I get that proton. But remember, I want the protons to be on this side of ATP synthase because they need to be able to go through that top part. Well, what happens is I have these awesome electrons, and not to get super science scary, the electrons, they fall from a high to a low state. And when electrons, let's put it over here, when electrons fall from high to low, that gives me energy. And what do protein pumps need over here to run? They need energy. So when this NADH comes up and breaks apart into NAD and protons and electrons, those electrons stay here in the protein pump. And what they do is they fall. And when they fall, that gives me energy. And that energy is needed to pull this proton up to the other side. So now I can start putting protons up here. And when I have protons up here, then they go through my ATP synthase. This thing spins. He takes my 2 substrates, the ADP and the P. He hugs them together. That spits out ATP. Okay, that's it in a nutshell. So now let's draw a really big picture, okay? So this whole picture, let's label it the electron transfer chain, which you guys should be kind of familiar with that word from one of your other biology classes. That's my ETC. So let's start by drawing some of those some of these protein pumps. So there's one, here's another couple, and I'm going to draw phospholipids here in a second. And then here's another one. Here's another guy. And then there's one more. So if we named these, this is complex 1. His name is Q, 2, 3, C, and then 4. Don't worry about the names right now. I just wanted to go ahead and name them because they do have names. And this is my cristae. So we need to draw phospholipids. Whoops, that's wrong. Draw phospholipids in the middle of it. So that way we know it's a membrane. Draw a couple more over here. There you go. All right, so let's label everything super fast. This guy right here, this is my cristae. This side of my membrane is the inner membrane space. This side over here, this is my matrix. And then, to finish it off, here's ATP synthase right there, all right? So let's do this whole reaction. The first thing, here's my NADH. We're going to talk about in another video where he came from. But the NADH goes up to that proton pump, or that protein pump, and he breaks apart into NAD, protons, and then my electrons. All right, so then what happens is these electrons, they fall from a high to a low state. So they just kind of start jumping to these proton pumps. So they fall from a high to a low state. When they fall from a high to a low state, that's going to take this proton and pump him through. So I start to accumulate protons on this side of my membrane. Okay, I put 3 up here, so it's not like you get 3 protons from your NADH. I'm just showing you that you're starting to build up. And then I actually have another friend, a lot like NADH. His name is FADH. FADH does the same thing. FADH goes up to the complex. He breaks apart into FAD, protons, and then he also leaves electrons. And so then these electrons start to fall from high to low states. A second, this guy can go over here too, so he just keeps falling down. As they fall down, they're going to create energy, which is going to take this proton, and it's going to go through that protein pump, and then I'm going to start accumulating more hydrogen ions on this side of the membrane, which is good because I want them to go from high to low. So I start getting those there. Now that I have these protons over on this side of my membrane, then they want to go from a high to a low state because they want to reach equilibrium, and they start to go through this ATP synthase. When they go through ATP synthase, that makes this top part spin, right? And when he starts to spin, then he can take my 2 substrates, let's make them something different. So here's my ADP, and then here's my other substrate, my inorganic phosphate. They go and fit inside the active sites. Well, as this guy over here is spinning, that is going to facilitate the reaction and shove those 2 really close together. When those 2 get shoved really close together, that creates my ATP. All right, so we just have to address one last problem. Because remember, all of these electrons are falling from a high to a low state, right? But that doesn't mean the electrons leave. They actually start to build up in this last protein pump. But if I keep building up electrons over there, are they going to be able to continue to fall from high to low? No, because this last one is full. So I need somebody to take them out of it. The guy that takes them out, well, that is not a good color. The guy that takes them out of this last protein, this is oxygen. And what have we said about oxygen this entire year? That oxygen is super greedy. And since he is super greedy, he likes to steal things. The things he is stealing now are electrons. Remember, he is just the thief all the time. And so when oxygen is sitting here in the matrix, he sees these free electrons, and let's find a different color. He sees these free electrons, and he starts to pull the electrons out. And when he pulls that electron out, he also sees, because like now he's looking over here, and he sees protons as well. And so when I have my oxygen, and I take electrons, and I see a few protons, so now it's oxygens plus hydrogens plus electrons, which acts like glue. Well, what do you get when you combine oxygen and hydrogen? You get H2O. And that's why whenever you guys breathe out and you go, and you do that on glass, and you can see where there's condensation, and you always draw little hearts, or you draw your initials, whatever the case is, that water is coming from this reaction, from oxygen taking electrons out of this electron transport chain and creating water. That way, this pump, this very, very last protein pump over here, stays empty, so the electrons can fall from high to low. Now, this entire process, this is going to create a proton gradient, because I'm shoving them from a high to a low, and then they fall back. This is a pretty special proton gradient because I'm doing chemiosmosis, right? So just kind of remember that word. Say it's a proton pump, or it's a proton gradient to make ATP. And so now, whenever we used to say the mitochondria is the powerhouse, that's fine, but we don't actually get to say it anymore because now we know what the mitochondria is actually doing. The mitochondria is doing oxidative phosphorylation, oxidative phosphorylation by ATP synthesis, which actually makes a lot of sense because oxidative, that's oxygen. I need oxygen over here so he can pull the electrons out. Phosphorylation just means I add a phosphate. Well, and that's what I'm doing right here at ATP synthase, is I'm adding a phosphate to my ADP. And then ATP synthesis, you already know what that is. That's creating the ATP because of it. So I'm doing ATP synthesis by oxidative phosphorylation because the electrons get donated by the NADH and the FADH. That gives us the protons and the electrons that fall through all these complexes. As they fall from high to low, that creates energy. That energy pulls the protons to the other side, to that inner membrane space. Now that I have the protons on the other side in a high concentration, they start to fall through my ATP synthase, which makes it spin. When it spins, it can take my two substrates, the ADP and the P. He can facilitate that reaction. I get ATP in the end. And then I need oxygen to pull the electrons out, so that way the electrons can keep falling, and that's why I get to make water. All right, that is how we make ATP. This is your electron transport chain. I hope that helps.